Looking at our next set of blood elements that it's part of a hematologic system, we get to platelets. Platelets are these fragments of larger megakaryocytes. So a stem cell, again, a hematopoietic stem cell or a hemocytoblast, differentiates into a megakaryoblast or a stage 1 megakaryocyte that then differentiates into a stage 2 and 3 megakaryocyte, and lastly, a stage 4 megakaryocyte. And then those little tiny components break off and they become our itty bitty little platelets. And so platelets, they have this blue staining outer region, they have purple granules, and they contain chemicals that support our clotting process. Things like serotonin and calcium and ADP and platelet-derived growth factor, amongst other enzymes. Their function is to form a temporary platelet plug that helps to seal breaks in blood vessels. And platelets are kept inactive and mobile by nitric oxide and prostacyclin that's secreted from our endothelial cells. They do have a contractile capability and they contain actin, myosin, and thrombostatin. And they have a residual ER, endoplasmic reticulum, and Golgi apparatus, which can synthesize these components. The mitochondria enzymes do produce ATP or ADP. Platelets on the surface have a glycoprotein that repels an endothelium that's intact or whole, but it adheres to an injured endothelium and collagen that's been exposed, so it's relatively smart. It also has membrane phospholipids that can activate the blood clotting cascade. Platelets are regulated by thrombopoietin, kind of like erythropoietin, and they're formed in the myeloid lineage, same as erythrocytes, but they're from different cells. They're from megakaryoblast, and mitosis will occur with cytokinesis until we get this large stage 4 megakaryocyte that has a multi-lobed nucleus. And then that cell will send these cytoplasmic components or projections into the lumen of the capillary, and then they break off. And these projections that break off are the platelet fragments. Platelets can live, or their developmental cycle takes about 10 days. And then once they're in the blood, their half-life is around 8 to 12 days. This shows all the formed elements of the blood. This is a great study slide. Thus far, we've learned about erythrocytes here and platelets here. Come next week, we're going to learn about all of our different leukocytes as well.